Students of B.Sc 3, Semester 5, CBCS syllabus, paper number 11 is Cytology and Research Techniques in Biology. Unit number 2 is Cell Organelles. In today's lecture, we will see about chloroplast and ribosomes. First, we will learn about cell organelle chloroplast. Chloroplasts are present in all green plants and absent in animal cells. It captures the sunlight and transfer it into chemical energy. Chloroplast is used for energy transformations in cells and living system. In the eukaryotic plant cell, photosynthetically active plastids with green colored pigments are called as chloroplast. Photosynthetically non-active plastids other than green color pigments are called as chromoplast, and colorless plastids are called as leucoplast. These three types of plastids are interconvertible and can transform into one another. Now we will study about shape, size and number. It shows variation in size due to environmental conditions. In higher plants, size of chloroplast is 4 mu to 8 mu in diameter and 2 mu in thickness. Its shape is spherical. In lower plant, it shows variation in the size. Chloroplastid in shade plants are larger in size than those in sun plants. Cup-shaped chloroplast is present in Chlamydomonas, spiral in Spirogyra, star-shaped in Zygnema. The number of chloroplastids is a genetical character, therefore it is constant. Higher plants show 20 to 40 chloroplasts and lower plants show few chloroplasts. One chloroplastid in Spirogyra and Anthoceros, 2 in Zygnema. See this in figure. Cup-shaped chloroplast is present in Chlamydomonas, spiral in Spirogyra and star-shaped in Zygnema. Chloroplast may be agranar, that is without grana region, and it may be granar when grana and stroma regions are present. In both type of chloroplast, envelope is present. In this slide, we will study about origin of chloroplast. It may be originated from pre-existing structure. In monotropic development theory, plastid are useful to produce the chloroplast. Development from proplastid and initials. In the cytoplasm, plastid initials are present in form of matrix. In the initial stage of development, the inner layer of membrane start folding. The folds are projecting inwards parallel to the surface of the initial, and such initial produce proplastid which develop into chloroplast. Development by division and budding in existing plastid occurs and new chloroplasts are formed. Development by nuclear division is one method of origin of chloroplast. The region surrounding nucleus and evagination process of nuclear envelope shows presence of initials which later on develop into proplastid and chloroplastid. Biogenesis in chloroplast. It involves the origin and development of chloroplastid in cell of cytoplasm which is developed from the double-layered proplastid with 20 nm size. In presence of the sunlight, it increase their size and become 1 mm. Invaginations of the chloroplast form lamellae of the chloroplast. Light absorbing pigment is pigment phytochrome. When light is absorbed, the invagination of inner membrane of proplastid forms vesicle, and during maturation it forms thylakoids. This figure represents biogenesis of chloroplast from proplastid. Then we will see about structure of chloroplast. It consists of outer envelope and inner matrix. This layer is without chlorophyll and cytochrome, but it is useful protective covering and region of the molecular exchange between cytoplasm and chloroplast of matrix. The matrix of chloroplast is gel fluid containing 50% of chloroplast protein. Stroma region shows ribosome, DNA and machinery for protein synthesis. In photosynthesis process, all the reactions in CO2 fixation takes place in stroma region. The stroma region is able to do synthesis of starch, protein and sugars. This region shows lamellar system. The lamellar system develops from vesicle present in stroma from inner membrane of chloroplast envelope. The lamellar system embedded in the stroma shows thylakoids, which are flattened vesicles in membrane network form. Thylakoids are piled one above other like stack of coins forming grana region, while in other region they may be unstacked. The lamellae are the stroma thylakoids forming system of tubules. Grana region are perpendicular to the surface of chloroplast and stroma thylakoids are parallel to the surface. This figure shows structure of chloroplast. It is with outer membrane, inner membrane, stroma lamellae, grana lamellae, starch grains, lipophilic globuli which are present in the chloroplast. Three-dimensional view of grana thylakoid shows grana region and stroma region. Chloroplast membrane is envelope containing two chloroplast membranes separated from each other by 10 to 20 nm. The membranes are 60 nm, made up of lipid, protein complex molecule and plasma membrane. Outer membrane is smooth and in direct contact with inner membrane which produces the vesicles. This membrane is with high selective permeability to the molecules. The lamellar system in stroma region consists of thylakoids, vesicles forming membranous network. This system provides maximum surface area for phytochemical reaction during photosynthesis. In this slide, figure represents thylakoid showing lipid, protein, chlorophylls. A thylakoid in this laminar system in a grana region shows double layered envelope. The space between the thylakoid envelope is called as interthylakoid space. Each thylakoid is made up of lipid and protein. Proteins are intrinsic or extrinsic. Intrinsic proteins are embedded deep in the lipid layer and extrinsic protein move on the thylakoid surface. The proteins are required for light reactions because protein chlorophyll molecules dispersed within the thylakoid membrane. In grana region, upper surface of inner membrane shows presence of small particles, contasomes. These contasomes are photosynthetically active pigments essential for photochemical act like absorption of light. Cytochromes, coupling factors and ATPase enzymes are used for conversion of fixed energy into chemical energy. In this slide, we will study about macromolecular complex in the chloroplast. It is used in photochemical act and harvest the sunlight energy. It shows photosystem first, present at the outer face of grana lamellae with reaction center P700. Photosystem second, present at the inner surface of grana lamellae with reaction center P680. It shows carotenoid protein complex, chlorophyll A protein complex, chlorophyll B protein complex, light harvesting complex, LHCP complex is required to transfer captured energy to photosystem second. Chloroplast contains cytochrome F B6 complex, cytochrome F, cytochrome B6 and copper containing protein plastocyanin. This complex is involved in energy transfer or electron transfer process during fixation of light energy and synthesis of ATP or adenosine triphosphate. Coupling factor complex CFO or hydrophobic protein complex HFO is involved in proton transfer in photolysis of water. Photolysis means breakdown of water molecule. CFO helps in binding coupling factor 1 CF1 to form ATPase enzyme. Such type of macromolecular system is present in the chloroplast. Photosystem first, photosystem second, light harvesting complex LHCP, cytochrome F B6 complex are organized in the contasomes in the grana region of the chloroplast. Coupling factor complex and coupling factor 1 CF1 molecules are present in the outer surface of thylakoid membrane in the region of stroma thylakoid. CF1 attached to the membrane with the help of CFO. The electron acceptor substance are present in the outer surface of thylakoid membrane and electron donor substances are present inside interthylakoid space in the grana region. Macromolecular complexes within the chloroplast thylakoid membrane are helpful in process of ATP synthesis during photophosphorylation in the photosynthesis. After this study, we will see about the second cell organelle. First, we will start with an introduction. The second cell organelle is ribosomes. Ribosomes are ribonucleoparticles found in all cells and assembly shops of protein synthesis. They are dense round granules found in cytoplasm of eukaryotic cell or attached to the endoplasmic reticulum. It was discovered by Claudi and named as microsomes. Later on, Pallady discovered and named it as a ribosomes. Robertson named it as a ribonucleoprotein particles. Ramkrishnan, Stills, Yonath described the function and structure of ribosomes. E. coli bacteria consists of 30% of the cell mass and 1 mm liver contains 2 into 10 raised to 13 ribosomes. Ribosomes are hydrated and 100 angstrom in height and 150 angstrom in the width. Its diameter is 23 nm and these are made up of 40 to 60% of rRNA and protein particles. They are present in clusters and held together by mRNA strand or string, and they are called as a polyribosomes. Look at this figure. Polyribosome formed by 5 ribosomes and mRNA molecules. Let us see structure of ribosomes. It is rounded 23 nm particle of large and small subunits. These particles are donated by their sedimentation coefficient. S. S is a Svedberg unit for sedimentation coefficient, which is related to molecular weight of the ribosome. Eukaryotic cell ribosomes are with sedimentation coefficient ATS when both subunits are together. When the subunits dissociates, the large subunit is 60S and small subunit is 40S. Prokaryotic ribosomes are smaller and associates at 70S and dissociates at 50S and 30S. Ribosomes in the mitochondria and chloroplast are smaller than ATS. Large and small subunits of ribosomes 40S, 60S, and when it is associated, it forms ATS ribosome. This slide shows large and small subunits of ribosome showing slots. Large and small subunits of ribosome shows slots. It shows 3 binding sites, A, P and E site. A site carries tRNA containing activated amino acid. P site carries tRNA containing polypeptide chain. And E site is the exit site from where the deacylated tRNA is released into the cytosol. In eukaryotic organism, only site A and P are present, while E site is absent. The binding sites are contributed by both the ribosomal units. Now we will see about fine structure of ribosome, which shows smaller subunit with head, base and platform. It was studied by Stoffier, Wittmans and Lex. The platform separates head and base with cleft. Large subunit shows ridge, central protuberance and stalk. Ridge and central protuberance is separated by valley region. Ribosomes can be sedimented by centrifugation of cell homogenate at a speed of 1,05,000 into 9. It is not membrane bound body without lipid and it consists of rRNA and protein. The proportion of RNA and protein is 1:1 in higher plants, while bacteria shows 2:1 RNA and protein proportion. These are strongly negative binding cations and basic dyes. See this figure. 3-dimensional model of ribosome with its functional sites. Smaller subunit shows head, platform, cleft, base. Larger subunit shows ridge, valley, protuberance. And when it is get associated, it shows tunnel for amino acids. Prokaryotic ribosomes contains 16 sRNA and 21 proteins in smaller subunit, while 23 S and 5 sRNA with 34 proteins in larger subunits. Eukaryotic ribosomes contains 18 sRNA with 30 proteins in smaller subunits and 28 S, 5.5 S, 5 S with 40 protein in larger subunit. In prokaryotic and eukaryotic cells, that is in simple and in advanced cell, out of total RNA, 80% RNA is rRNA. If MgIn concentration in the cytoplasm is lowered, the two subunits fall apart and get separated, while increasing concentration by the addition of MgIn helps the association of subunits into formation of the complete ribosomes containing proteins at some definite sites of polyribosome formation. This figure represents composition of prokaryotic and eukaryotic ribosomes. It shows different type of sRNA and proteins in prokaryotic and eukaryotic ribosomes. rRNA has high degree of secondary structure. Nearly 70% of rRNA are double-stranded and helical due to the base of the same RNA molecules. It provides three-dimensional matrix to which protein synthesis enzymes bind in definite order. In prokaryotic ribosomes, 16 sRNA in smaller subunit has a sequence at its third end complementary to the ribosome binding site of the mRNA, which is useful in polyribosome formation. In prokaryotic ribosome, 5 sRNA in the larger subunit has a sequence complementary to a sequence in all tRNA, and it is used for binding of tRNA with ribosome. Ribosomal RNA provides some catalytic activities for protein synthesis. In mitochondria, ribosomes synthesize protein required for the enzymes, cytochrome oxidase and ATPase, while chloroplasts have their own DNA, RNA and ribosome for the protein synthesis. In this slide, we will learn about functions of the ribosomes. Ribosomes are involved in protein synthesis. When not engaged in protein synthesis, ribosomal subunits are separated and exchangeable. The ribosomes are separated by dissociation factor, which is the initiation factor for binding of mRNA to 30S subunit of ribosome to start the protein synthesis. During protein synthesis, many ribosomes attached to mRNA molecule to form polyribosome. The structure of the ribosome shows that after binding of two subunits, a groove exists between two subunits, and in the groove, mRNA molecule is located. As the mRNA molecule remains in the groove, the ribosome protects a stretch of 25 nucleotide of mRNA from degradation of ribonuclease. The structure of ribosomes is suitable for elongation of polypeptide chain. The large subunit shows tunnel and developing peptide chain grows through this tunnel, due to which ribosome protects a segment of about 30 to 40 amino acids from degradation of proteolytic enzymes. Ribosomal subunits dissociate after protein synthesis. And when there is a protein synthesis, ribosomes get associated with each other and forms tunnel-like structure. Figure represents ribosomes in polyribosome showing portion of mRNA, tRNA and tunnel in polypeptide chain. The main role of ribosomes is in protein synthesis. Ribosomes are attached on mRNA string, forms amino acid pool. Then through transcription process and translation process, there is a formation of amino acid chain and polypeptide structures, peptide bond structures, and then there is a protein synthesis. Transcription and translation, these are two important processes present in protein synthesis. We will see detail about the protein synthesis process. It starts with the binding of small ribosomal unit to mRNA binding site, which contain AUG codon to signal the start of codon. The other factors present in the cytoplasm are involved in bringing the correct amino acid tRNAs to ribosomes. In ribosomes, amino acids are linked in a sequence to form polypeptide chain, which results in the protein synthesis. This activity is controlled by genetic message carried in nuclear DNA. It is transcribed manner mRNA contain coded message for protein synthesis. Two subunits of ribosomes are unequal and perform specific function. Ribosomes are required for synthesis of large protein molecules. Ribosomes are arranged and linked by thread of mRNA. The functional group of ribosomes are called as polyribosomes. The mRNA produced is translated according to genetic code into sequence of amino acid in the polypeptide. The mRNA is absorbed on ribosomes. Two ribosomes contains 35 RNA molecules and 50 different proteins. Transfer RNA are involved in transfer of amino acid from cytoplasm to ribosomes. It shows two steps. First, amino acid molecule is activated by specific activating enzyme to react with ATP. This forms amino acid adenylic compound and release of pyrophosphates. Amino acid adenylic monophosphate is then attached to specific tRNA present in cytoplasm and transferred to ribosomes. Single-stranded mRNA is attached to the smaller subunit of ribosomes. It contains information of amino acid sequence. The sequence of nucleotide on mRNA forms codon. For each codon on mRNA, there is a complementary anticodon on transfer RNA. This anticodon recognizes complementary codon on messenger RNA. The formation of polypeptide chain shows three steps as initiation of polypeptide chain, elongation of polypeptide chain and termination of polypeptide chain. In second step, ribosome protect mRNA strand which passes between two subunits of ribosomes from the action of nuclease enzymes. It also protects polypeptide chains passing through the channels between the subunit against the action of proteolytic enzymes. So this was about protein synthesis process, which is related with ribosomal subunits, smaller subunit, larger subunit, mRNA, tRNA. Bibliography, different books to perform these notes. Long answer type questions and short answer type questions from this topic. Thank you.